transitioning now away from the systems approach to look at a more cellular thing. We're going to start with Carmen Canavier. She's from LSU. She's spent her career modeling the firing properties of dopamine based on their intrinsic properties. And she's going to talk to us about that right now. Cameron, carry on.
Here we go. Okay, can you hear me? Yes. All right, so thank you to the organizers for inviting me. Thank you to John for that nice introduction. Thank you to the National Institute on Drug Abuse for funding this work. As John mentioned, it is going to be a departure from the talks where they were talking about what does dopamine mean. I'm going to talk more about the intrinsic dynamics of dopamine neurons themselves. The title is What Constrains the Maximum Firing Rate in Midbrain Dopamine Neurons. This is joint work with Chris Knowlton in my lab and Jochen Roper in Frankfurt, Germany. Okay, next. In vitro, dopamine neurons are slow, regular pacemakers. So everyone's been talking about in vivo, but I'm going to talk about their intrinsic properties, and they are slow pacemakers. So depolarization block has been known to limit the firing rates of these neurons. In this paper from the Katai lab. So they're pacemakers, and as you depolarize them, their frequency increases, but only up to a maximum of about 10 hertz. So this is in the context of Tony Grace's work from the 80s, where tonic firing rates in these neurons in vivo are about 1 to 8, and then any faster than that's burst firing. So this has some implications. If they can't fire any faster than 10 hertz, then how do they fire faster in vivo? And the answer likely is the synaptic integration of the synaptic inputs. So that data that I showed you was from substantia nigra. And originally, VTA dopamine neurons were thought to be homogeneous and conventional like the nigra neurons. And this is a review from Mark Unglis and Tony Grace, summarizing what was thought about the VTA neurons. And they identified them with TH. And panel B shows that they have a prominent H current. This is under voltage clamp. Under current clamp, it would manifest itself as a sag. They're slow, regular pacemakers, as I stated, and they have a relatively broad action potential. However, it turned, as acknowledged in that review, most of the neurons that this view was based upon were in the substantia nigra and in the part of the VTA that projects to the lateral shell of the nucleus accumbens. So Stefan Lammel in Jochen Roper's lab in 2008 did a systematic study where he labeled these neurons by their projection targets. And these are the major results, in that he did find the conventional neurons as stated, and this is what the procedure here was. He identified the neurons as being TH positive. He also injected beads into the projection area so he can separate whether they're mesocortical, mesoamygdala, mesolimbic core, or whether they project to the medial shell or the lateral shell of the nucleus accumbens, or whether they're neostriatal. And so he did find this conventional population, and what he's done is he hyperpolarized them, then gave them a triangular ramp in current. And you can see this on the left-hand side, it's a weak ramp on the order of 50 picoamps. On the right-hand side, it's a stronger ramp, 1 to 200 picoamps. And then for the stronger ramps, they go into depolarization blocks. So they fire on the up ramp and not so much on the down ramp. And same for these, the VTA cells projecting to the lateral shell. But the rest of the VTA shells have a different electrophysiological profile. So you can see over here, under these conditions with the triangular ramp, the maximum rate is about the same as it was in the earlier study with a square pulse, and it's about 10 hertz. But the other population of dopamine neurons, which was called atypical in that paper, including the cortical projecting, amygdala projecting, and the core and medial shell of the nucleus accumbens projecting, they have a faster basal firing rate, and they have a faster maximum firing rate. So they can fire on the order of 20 to 30 hertz. And you'll also notice they go into depolarization block in a different manner. So the amplitude trails off as opposed to an abrupt failure in the atypical versus the conventional. And I'm going to focus on two just templates or exemplars of this atypical electrophysiological profile that's faster firing and fails at a faster frequency in a different way, and this conventional profile. This one's from a nigrostriatal neuron, and this is from a mesolimbic projecting to the medial shell. So we're just going to focus on these as exemplars of how they go into depolarization block. And we're going to use a simple one compartment model to capture the mechanism of depolarization block that limits the firing rate. So just a simple model. It's a capacitance in parallel with a sodium current for the upstroke of the spike with a leak conductance with 3 potassium channels, including a delayed rectifier, the A current, and calcium activated small conductance calcium channel, 2 calcium currents, the N and the L, and the H current. We also, in addition to the equivalent circuit model, we have a model of the accumulation of calcium so that the calcium entering via the calcium channels can activate the calcium activated channel. Now, we also know from papers from Vince Sutton and Dominic Engel, and also from this Ding et al. Paper from 2011, that there's a slow cumulative activation of the sodium current in dopamine neurons. And this is not to be confused with the fast inactivation of the Hodgkin-Huxley model. Instead, if you take nucleated patches and you clamp them for a brief period of time to 0 millivolts, and then you let them recover for 100 milliseconds in between pulses, you get a decrement in the recorded sodium channel current due to this very slow process of, slow recovery. It inactivates rather quickly, but the recovery from this slow inactivation is on the order of 100 milliseconds. So Chris Knowlton in my lab calibrated the sodium channel, and we used a Markov model with 1 open state, 2 closed states, a fast inactivated state, and a deeper inactivated state we call the slow inactivated state because it recovers slowly from this slow inactivated state, based on a paper from Balbi et al. 2017. And he used the exact protocols from the Ding et al. 2011 paper for activation, the voltage clamp protocols to measure activation, and the voltage clamp protocol to measure fast inactivation. These protocols are so brief, they're not going to measure this any occupancy in the slow inactivated state. And so the red line is the best fit to the Ding data. The triangles are the, this is the inactivation fit that Chris got. This is the activation curve from Ding et al. And this is Chris's, in his activation curve. This is one concession. We did say this is a 1 compartment model. And Chris wanted to honor the fact that dopamine neurons have a depolarized spiking threshold of about -40 millivolts. Therefore, since we don't have an axon initial segment in the model, he had to make the activation of these sodium channels steeper than is actually observed in these somatic outside-out patches. So our modeling approach was, first, we were going to calibrate an atypical model for the ventral tegmental area, less the lateral shell projecting neurons, because it's actually easier to get that failure mode, to get that kind of depolarization block. And then we were going to identify which parameters in the model are critical to switch between the atypical and conventional electrophysiological profiles. And finally, we were going to come up with our final calibrated conventional model for the nigrostriatal cells plus the lateral shell projecting cells, and make testable predictions regarding these 2 types. So there's known differences between the, known biophysical differences between the atypical population and the more conventional population. First and foremost, as I stated earlier, the conventional cells, if you hyperpolarize them, they have a sag current response due to the H current, the HCN. And whereas in the atypical, this H current, the sag is either absent or weak. They have a slower recovery from inactivation of KV4 of the A channel. And this is manifested in, there's a longer ramp, a longer time before you start spiking after hyperpolarization, because you want to recover, you need to recover from an activation of the A channel. And this different time constant, which would be 5 or 6 times as large in the atypical as the conventional, was documented first by Cassiotis from the Roper lab in an SFN abstract in 2011. And more recently, Zaid Khalik's lab has published Tarfa et al., in which they showed that the time constant of recovery from inactivation is rather, fairly fast in the nigra compared to the mesoaccumbal, although there's a big spread in the mesoaccumbal, and we believe this is because it includes the more conventional cells projecting to the lateral shell of the nucleus accumbens, as well as the atypical population. So another point is they have a deeper AHP in the conventional, deeper after hyperpolarization in the conventional as compared to the typical. So we started off with our atypical model, not putting any HCN in it, making the time constant for inactivation, which is also the time constant for recovery for inactivation, of KV4 to be rather slow. And we used actually rather small values for the delayed rectifier and the calcium activated SK in order to produce the weaker AHP. And finally, these cells the atypical cells are somewhat smaller than the conventional cells. That's based on some data from the Lammel et al. 2008 paper, but also some unpublished data from the Roper lab. All right, so the next step is we're going to calibrate our atypical model on basal rate and action potential shape according to data from the Lammel et al. 2008 paper. So our atypical model paces relatively quickly compared to a conventional one. It has a smaller and wider action potential shape, and this is reflected as a small excursion in the phase plane of the derivative of the membrane potential of the action potential during the action potential and the actual potential. So in the phase plane, we also, so Chris calibrated the model to match the known data on the atypical population, and then applied depolarizing ramps to try and match the data on the response to triangular depolarizing ramps and the entry into depolarization block. And if you ignore any entry into the slowly inactivated state, a weak pulse does not induce depolarization block. A strong pulse does induce depolarization block, but contrary to the experimental data, if you ignore entry into the slowly inactivated state, you emerge out, you start spiking and don't remain in depolarization block on the downstroke of the ramp. So there's no hysteresis, and you need this slow process in order to get the hysteretic effect that you spike on the up ramp, but you don't spike on the down ramp because you're in depolarization block. What's shown at the bottom here is occupancy in the I1 state, meaning the fast inactivated state. And the reason you go into depolarization block is basically as everything goes into this I1 state and you don't have any current available to generate another spike or depolarization. Now, so the next thing that Chris did was he added slow inactivation, some occupancy into the slow inactivated state in order to reproduce the hysteresis. And so he then gets no depolarization block with a weak ramp, entry into depolarization block at about the right frequency and with about the right shape, in that both the AHP and the peak of the action potential just sort of trail off into depolarization block. Now, I plotted additional things at the bottom here on the occupancy of the state. That's still the occupancy in the I1 state is blue, but now we show the occupancy in the I2 state in green. And we show the red is a little bit more complicated, but it's important. It's of this available pool, meaning all the neurons that are not, all the channels that are not in the slowly inactivated state are available. And what the red shows is what fraction of those channels that are available are in the fast inactivated state. So when they all go to the fast inactivated state, you again get depolarization block here. All right, so we then next checked. I previously said there's known biophysical differences between the typical and atypical, and the atypical and conventional populations. So we tried each one of these differences individually and then combined, and none of them had an effect on the maximum frequency. The peak, all these manipulations, the peak frequency stayed around 30 hertz. The shape stayed the same, so increasing the H channel conductance just allows you to spike a little bit sooner. Speeding up the time constant for the KV4 current just allows you to spike a little bit sooner. The failure is about the same. Increasing the two currents that contribute to the AHP, the delayed rectifier, and the SK channel, again, did not have a lot of effect. And changing everything together did not change the mode of failure, didn't change the rate at which it failed. It just allowed it to spike a little bit longer. right, so then we asked ourselves, if none of the known differences can account for the different electrophysiological profiles of the two populations, what does account for it? And so Chris increased one parameter, and that was the rate, the scale factor for these are all voltage dependent rates or transitions in this Markov model, but he increased the scale factor for the transition from I1 to I2, so that it's much more likely to go into this inactivated state in the conventional versus the atypical, and just that one parameter change immediately caused a limitation in the maximum firing rate from 30 to 8 hertz. And it also, without planning, and it also just automatically switched to this different mode where you get a few spikes and then a sudden failure. So that made us think we're on the right track because we just changed one parameter and we got this for free. And another, but then this is not the final refined model. This is just a slide that shows, aha, this is the critical parameter. Because when the cell goes into depolarization block with too little current, it spikes too little. And so we're going to continue to fine tune this model. But the bottom line is you have, and we'll return to this point, but basically in the atypical failure mode, you basically have everything in this I1 state. But in the conventional failure mode, you do have some availability, some occupancy in the C1 and C2 states. And so this is contributes to the different mode of failure. So I said we were going to recalibrate the conventional model now that we have a calibrated atypical model, and then we're going to go back to the Lammel data. So the conventional population fires more slowly. It has a taller, narrower action potential, and this causes a larger excursion in the phase space of the derivative, which is proportional to the current, and the membrane potential. So which parameters did he have to change? Basically, we not only need more slow inactivation of the sodium channel in the conventional population, which is the striatal, to remind you, the striatonigral plus the lateral shell projecting cells in the VTA. But we also needed to strengthen the spiking currents to get a good correspondence. So we needed to increase both the delayed rectifier and the fast sodium channel. And also we needed to increase the sodium, to get a good fit, we needed to increase this rate of entry into the slowly inactivated state even more. We've multiplied by a factor of 4 here as opposed to the 3 in the previous slide. And we made a little change to the kinetics in the delayed rectifier. We shifted the time constant a little bit to the right. So this is the final fit. On the left-hand side, there's the atypical versus conventional from the Lammel et al. Paper that initially defined these subpopulations. And on the right-hand side is the final model simulations of the atypical, which is fast firing. The conventional has a slower spontaneous firing rate. And we reproduce the shorter, wider action potential in the atypical population as opposed to the taller, narrow one. And we also got a qualitative fit for the larger excursion in the phase plane. So this is kind of the punchline in that now we have an atypical model that it does not go into depolarization block with a small pulse. It does go into depolarization block at the correct frequency and with a qualitatively correct shape for a strong pulse. And the conventional model does not go into depolarization block with a weak pulse, but it does go into depolarization block, and it's a different depolarization block because it's a very abrupt failure of spiking at a much slower frequency.
We've got a couple more minutes.
Okay, well, I'm going to skip right ahead then. Like 2 minutes? Okay, so this is a head-to-head comparison of the triangular pulse data. So at the top here, this is what I showed you, the exemplars from Lammel. That's the atypical compared to the model. This is the exemplars from the conventional compared to the model. So we feel like we have a decent qualitative fit to the failure mode. I'm probably going to have to skip over this pretty quickly since we're at the end, but I did want to revisit the square pulses from Richards that I started the talk with. And so you give a square depolarizing pulse, you get the same, very similar type of failure as we did to the ramp. Similar with the atypical, you give it a pulse, a depolarizing square pulse, it goes into depolarization block. This is actually, to my knowledge, this experiment has not been performed, but we predict that the atypical will go into depolarization block the same way with in response to a square pulse as to a triangular pulse. But again, this is an important distinction. There's still this red line is not at 1, meaning that not everything is in the I1 state. whereas in the conventional, everything's in the I1 state, nothing's available for the next spike. And that becomes important because this is a model prediction. In the typical, if you're in depolarization block and you give it a little bit more depolarizing current, nothing happens. For the conventional, since you do have some sodium channel availability and you give it another pulse, you can actually cause more spiking. So this may be important in terms of integration of synaptic inputs. So conclusions are that the known differences, the known biophysical differences between the atypical and conventional populations do not account for the differences in their maximum firing rate or their failure mode. Conventional neurons fail at lower frequencies despite the fact that they have larger, we predict they have larger spiking conductances, and we know that they have deeper after hyperpolarization potentials because of the larger contribution of slow inactivation. You would expect that bigger spiking conductances and more AHP would actually allow you to remove more inactivation between spikes, which would allow you to keep spiking longer and not go into depolarization block, but that doesn't happen because of the larger contribution of inactivation. And finally, we predict two major differences, more inactivation, larger spiking touches. These differences can be tested with a dynamic clamp and likely impact synaptic integration. So future directions, we need to look at the effect of synaptic inputs of morphology and use a dynamic clamp perhaps to convert between typical and conventional cells. I'm gonna stop here acknowledging the collaborators.
Cool. Well, thank you very much, Carmen. I'm going to turn it over to Paulina, who will direct questions back to you.
Right. Thank you so very much. That was very interesting. I have a question here from Jan Rodriguez. So he's asking, is it possible to speculate how the differences in the spiking pattern would affect dopamine release?
Well, like I said, this is all, it's modeling of in vitro conditions. Like I said, one prediction would be, and actually I'd like to elaborate a little bit. So the basic idea is that you have to, this is entry into the inactivated state. And for all the activation, if the spike causes inactivation, then you have to remove it, and you have to remove the exact same amount that the spike caused by the time of the next spike, otherwise you can't maintain. That's really what's causing the failure here, is you're not recovering from an activation to the degree that you're causing it. And so, okay, so all the fast inactivation gets removed during the AHP, but the slow inactivation continues to be removed during the whole interspike interval. And just to kind of get back to the question of what does this matter about dopamine release? Well, Tony Grace had some papers in the 80s, and actually more recent stuff too, about depolarization block in response to haloperidol, into antipsychotics, that you'd get some, after 3 weeks of treatment with haloperidols, you would go into depolarization block. But that wouldn't necessarily, in this conventional population, it wouldn't necessarily stop you from firing in response to additional depolarizations or inputs. So, but in these cells, it would. So that's one. Thank you for asking the question, because I didn't really have time to go through this slide. But that's one possible implication for dopamine release.
Fantastic, thank you. Maybe we have time for one or two more questions. So Jose Chavez is asking, do you know if this atypical neuron that go to the medial shell is a VGLUT2-TH neuron, the same as the calbindin positive neuron that Dr. Roper described?
I wish that Stefan Lammel could answer that question, because I know he addressed that in his recent review, and I actually don't remember the answer.
Okay.
I'm a modeler, so I didn't do any of the experiments. It's only modeling, but it's a good question, and I'm pretty sure that, yeah, that answer is in the review, but I don't remember it.
Okay. We have a question from Paul Dodson. So he's asking, if you have two strong sequential inputs, would atypical neurons pause after maybe a single spike when they go into the block, or whether typical neurons burst into the second input?
Oh, that's a good point. I'm not sure. I might have to ask for clarification, but my colleague Paul Shepard, that I have published with before, and we were thinking, people normally think of pauses as being a hyperpolarization, but you can get a pause from too much depolarization if you're in depolarization block. And so I don't know, does that answer? I'm really not, I like clarification. I mean, you can get pauses in both of these neurons from depolarization block. But can you repeat the question to make sure I answered it? You're muted.
Paul's question is, if you have two strong sequential inputs, would a typical neuron pause?
Too strong sequential. Well, like strong enough to, yeah, I guess I'm not too, I'm like very brief sequential inputs. I mean, I guess, yeah, you wouldn't need to have one long one. You're right. I mean, it's a sequence of depolarizing pulses could also push it into depolarization block if that's the question. I haven't done the simulations, but I'm sure it can be done.
I think we are probably running out of time now, but all of the questions that have been asked here will be posted to the Slack channel so that they can be answered and possibly request for clarifications. Thank you.